It's Annabelle Ballesta. She's an interim researcher with a background in applied mathematics and computer science. She started her own group in 2018 and is currently heading the cancer systems pharmacology team at the Institut Curie in CERN. Her team is dedicated to the design of mathematical and statistical methodologies for the identification of innovative drug targets and the personalization of anti-cancer therapies. They develop detailed mechanistic models that are interfaced with bioinformatics and statistical algorithms to integrate large-scale data sets into therapeutic optimization and identify biomarkers to stratify patients. I'm very happy that you are here. Annabelle, the screen is yours.
Thank you. Thank you very much for the invitation. Can you all see my screen correctly?
Yes.
Thank you. Okay, so today I will talk about systems pharmacology. I will try to define this type of techniques, and I will try to show an example of how we can use that to actually optimize cancer drug combinations. To start with, so we are all aware of the area of precision and personalized medicine. And nowadays we have multi-type data sets that could be either preclinical, you showed very nice examples today, or clinical data that could be available in patient populations. And the idea is to translate those numerous data sets into some personalized cancer treatments. And one way to do that is to use bioinformatics. Here I will show how we combine machine learning with mechanistic models, which sometimes are called digital twins, to try to design personalized therapy. In a nutshell, the idea is to design a mathematical model here, or let's say a framework, a bioinformatics framework, that would encompass all prior knowledge you could have on the specific drug or cancer type. And basically, you would just recalibrate this model using patient data, and then use that framework to design personalized combination chemotherapy. Or it could even be used for immunotherapy, for instance. So just a few words about what is quantitative systems pharmacology. Those words appeared like around 2012, something like this, according to a PubMed search. And the idea is to design mechanism-based mathematical models that would represent the key regulatory networks and also the drug pharmacokinetics and pharmacodynamics, using a type of mathematical tools that we call ordinary differential equations, ODEs. And so an ODE would look like this. And the idea is that if you want to represent this reaction, for instance, A becomes B. So A could be, for instance, some protein that could be, for instance, phosphorylated. So this reaction will have a reaction rate here, K, which is typically in the unit of per hour or per day. And then you can actually translate that into here an equation. And if you provide the initial conditions, so A at time 0 and B at time 0, you could actually compute the whole trajectory of the concentrations here. This is, for instance, protein concentrations or drug concentrations. So this is just for one reaction, but then if you do that for multiple protein interactions, or even you could actually go up to the level of organs or even whole body. So here, for instance, this is a model we developed for brain tumors, where you can have here the pharmacokinetics, the whole body pharmacokinetics, that is just given to some local model of the brain tumor with some different cell types, which could be healthy, like microenvironment, or cancer, like cancer cell populations. So why do we want to do that? So what could be the advantages of doing that? So basically what we can do is recapitulate multi-type data, which could be sparse. I mean, we don't, as compared to the statistics, we are less stringent regarding the data sets that we need. And the idea is that the mathematical model, as you can see here, the variables B and A, they do have a physiological meaning. So we could then make predictions. And very interestingly, those models are associated to a low computational cost, meaning that we can perform a massive in silico testing, and in particular, we can test millions of drug combinations in silico, or millions of conditions, for instance, gene KO. On the opposite, really the problem with this type, or the limits with this type of approach, is that for each reaction you have a parameter here, K, and you need to have enough data to be able to estimate this parameter, otherwise the model may not be accurate enough. And this is clearly what limits the size of the model. So here I will present an example which is under revision in the British Journal of Pharmacology. This is really the work of Sergio Corredore, who was a postdoc in my team, who is now at Servier. And this is also a collaboration with the Brain Institute in Paris, in Hôpital La Pitié-Salpêtrière. And I would like to cite Maïté Véro, who did the experiments for validation. So very briefly, probably most of you know the glioblastoma, but so this study is in the context of glioblastoma, so the most frequent and aggressive brain tumor in adults. It's associated to dismal prognosis, and it's a complex disease. Of course, it's very heterogeneous. It's also very plastic in the sense that it reacts, in particular, to the treatment, we know some type of the main drug against this type of cancer is temozolomide, which is associated with surgery and radiotherapy. And we already know some very famous resistance mechanisms, the most famous one being the repair by the MGMT enzyme, which is already included in the decision tree for clinicians. And I'm going to talk about this a bit more. So here the idea is to investigate temozolomide pharmacology and try to find some drug combinations that would circumvent the tumor resistance. As I was saying, we are going to use quantitative systems pharmacology and machine learning. So the first step here is to design a mathematical model. And the idea is to, of course, use the prior knowledge that you have on the pharmacokinetics and pharmacodynamics of temozolomide. We have also included the pathways which contain the most numerous, or the largest number of mutated genes in glioblastoma patients, as described by the Cancer Genome Atlas. And we have also used results from several CRISPR screens on the patient-derived cell lines, showing that some hits are extremely important for resistance. And putting all this together, the idea was to first design a core model before personalizing the model, basically. So here is the upper part of the model, just to give you a sense of what we are computing. So here, temozolomide is here. Everything in yellow would be the pharmacokinetic part. So temozolomide is first convert into MTIC and then into the cation, which is the active species, and AIC, which is completely inactive. And the cation here will then react with the DNA to create DNA adducts. And the most important one would be the O6 methylguanine here, which are repaired by this MGMT enzyme. So as you can see, basically, if this works perfectly, then the drug action would stop here and the cell would be resistant. There are some other types of adducts which are repaired by the base excision repair. And then we continued downstream. So that's the whole model. So I described the upper part. Now going to the so the lower part, basically you have several steps of DNA repair, which I'm not going to explain, and then you have the double-stranded breaks, which trigger ATR phosphorylation. So that would trigger cell cycle arrest here, and p53 network towards cell death. I will not go through that, but let's say that here the cell cycle is clearly not well detailed, because the only thing that we need here is a marker of S phase. We want just to know whether or not the cell is in S phase, yes or no. So we did something quite simple, which is here. You can see it's a cosine, actually. It's a 24-hour cosine. And here, because of course that's always the question of how to relate intracellular networks to actual cell death. And we have taken advantage of a nice study by Harvard Medical School, showing that basically if you look at the P53 AUC, area under the curve, what you can see is that here, over time, we look at P53 AUC, and one line is equal to one cell. So this is those are single cell data sets. And a line is red when the cell died, and a line is blue when the cell is still alive. And as you can see, we can draw this type of curve, discriminating between the cells that actually died and the cells that are still alive. And we have used that and programmed this in the mathematical model to say whether or not the cell will die. So this is clearly just a result of the profile of P53. Okay, so now we have the model. And the next step is to parameterize the model. As I was saying, the parameter estimation is the key point here that will determine the accuracy of the model. And here we have 62 parameters in total. So we did two steps. The first step was more like a one-by-one estimation when possible, which is always the most accurate and the most reliable way to estimate your parameters. But then the step two was basically taking the 32 remaining parameters and making the most of datasets which were available in two cell lines. So basically in the LN229 cell line, which is a commercial human glioblastoma cell line. So the original one, the control one, is doesn't express MGMT, so it's sensitive to temozolomide. This is what we can see here. So here we are looking at the survival of the cells as compared to temozolomide concentration. And those cells were transformed to overexpress MGMT. So those are the resistant cell line, or the one I will call MGMT plus. And as you can see here, they are completely resistant to temozolomide. Just to give you an idea, the concentration of temozolomide that you find in GBM patients are around 20, so that would be around here. Okay, so then we take all these data sets, we put all that into a parameter estimation procedure, which I'm not going to explain. And basically what we get is a model that fits the data. I'm just showing some example here. So for instance, here, here we are looking at the data according to the dose of temozolomide. The dots are the data points, and the lines are the best fit of the model, taking all data together. And the two colors correspond to the two cell lines, so MGMT minus, so the sensitive, MGMT plus, the resistant cell lines. And so we have data for these guys. As I was saying, those are very important, the DNA index. We also have data for the double-stranded breaks. As you can see, we do have some good agreement. Then we also have data for P53, both total and phosphorylated at the CER46. And then we have data on the percentage of apoptotic cells for different exposure of temozolomide and also of survival. And as you can see, we have a nice fit of the model to the data. We have other fits I could show you, but the idea is just to well, give you an example. But so basically, we did other validation to make sure that the model was accurate. And so now we do have a model, and the idea is to use it for optimizing drug combination. So the way we have done this is to identify drug combination that would potentiate temozolomide for resistant cell lines. And we also kept the fact that we want to maintain the efficacy on the sensitive cell lines, which I guess is the easiest part. So we combine temozolomide with what we call targeted molecules, so that will inhibit one molecule or one pathway at a time. And we explore the inhibition from 0 to 100%. And then we look at the efficacy of temozolomide plus those molecules after 5 days. And basically, the output that we are looking at is temozolomide IC50 in the presence of the targeted therapies. So basically, here I show you two examples, but basically that would be this value here. So the concentration at which 50% of the cells are dead. So of course, the smaller, the better. in terms of efficacy. Okay, so we did that for all possible targets in the model. So here I have listed the targets. It's maybe difficult to read, but basically here we have the PK, here we have the DNA repair. And here on the x-axis, you have the percentage of inhibition of each target. So from 0% to 100%. And the left table is for the sensitive cells. The right tables are for the resistant cells. And basically what we want is low values of IC50, so yellow. And as you can see, so for sensitive cells, that's quite easy. For the resistant cells, we do have actually 5 targets that pop up. So those are the base excision repair activity, homologous recombination activity, ATR degradation, and phosphorylated ATR degradation, and p53 degradation. So that's the, I mean, just giving a single, let's say, combining temozolomide with a single targeted agent already achieves some good level of activity, and already at 40% of inhibition, which is something that sounds realistic in the clinics. Clearly there is I mean, 2 of them are better, the BER and HR, as compared to the 3 others. And then the next question was, okay, now it's just temozolomide plus 1 drug or 1 compound. What about temozolomide plus 2 compounds? So now what we did is we set basically the inhibition level to 40%. And then, yeah, I forgot to say that, maybe that's important. I forgot to say that inhibiting MGMT, which is a typical or the most straightforward way that any person would do that, is not completely realistic, because you basically, so that's us here, you basically need 100% of inhibition to achieve something close to what you would find in the optimal solution. So that's clearly not what you would go for if you look for optimal solution. Okay, so now going back to what I was saying. So basically we have temozolomide plus 2 drugs and also at 30% inhibition. And here, so this is a matrix, so basically 2 triangular matrices. So the lower part is for the sensitive cells, the upper part are for the resistant cells. And here we, again, we have the list of the targets that were inhibited in silico. And as you can see, so here, okay, no problem, and sensitive cells are sensitive in any cases. In any case, on the opposite here, you do have few yellow spots, which are the interested ones. And as you can see, they do organize into lines. And again, so here we can see the base excision repair and the homologous recombination here. And we also see the TP53, that sounds a bit interesting here. Okay, so basically now we have a few of them that are of interest. So what we did, we went one step further, and then for those I think those 3 ones, we plotted the results, the temozolomide IC50 with respect to the level of inhibition. So here I'm showing for instance, if we look at this one, this is the level of inhibition of the homologous recombination repair, and this is the level of inhibition of p53 degradation. And if you look carefully, you would see that this one, so we are looking for yellow, basically. And if you look carefully, you can see that here is flatter as compared to here, in the sense that here you need less inhibition, or a smaller inhibition, or less strong inhibition to achieve maximal efficacy. So this is how we decided that this combination would be the optimal one. So again, temozolomide plus 2 inhibitors of 2 repair pathways. Just to give you some mechanistic explanation. So again, this is the model, and what the optimization procedure suggests is that basically you need to forget about this path here, because the MGMT is basically too strong, or the overexpression is too strong, and then try to open this path by basically inhibiting that. So here you open that path basically, and here you inhibit that, so you open that path, and then you can go to the downstream cell death basically. And then so we validated this in vitro In MGMT minus and MGMT plus. So here, if we focus maybe on that, so the black line is temozolomide alone. And then if you give the optimal combination, which is the green one, you can see that you do have a very nice increase of temozolomide efficacy, and this is the best one as compared to the two other ones. Maybe I should say that orange is niraparib, is temozolomide plus niraparib being an inhibitor of base excision repair. This is written here, and RI1 is an inhibitor of HR. And I have two more slides. So the next thing that we did is try to combine this approach with machine learning to understand why this optimal drug combination is optimal and for which patients. So really in a nutshell, so the idea is that you use your model to design virtual patients. So a virtual patient is basically a parameter set, and then you use the model to compute this type of curve, so which are basically the sensitivity to temozolomide, or here to the 3-drug optimal combination. And then you use machine learning to design a model that is not really mechanistic anymore, but is just a black box, more or less, not really a black box, let's say gray box. You design this model, and then you ask to this model, okay, what was the parameter that was the most important to define the sensitivity to this? And here we actually said that we don't want the sensitivity just to the 3-drug. We want to have a differential effect between temozolomide alone and the 3-drug. So this is just taking the difference of the AUC. And so just to give you the results, so basically those are the parameters which are important for having a differential effect in the efficacy of 3-drugs versus temozolomide alone. And as you can see, MGMT level is very important. We also have the level of ATR, and then everything in black would be related to the apoptosis level. And we also have P53 that is of importance. And to use that for the clinics, the idea would be to relate these model parameters to some clinical biomarkers, which could be done. And this is what we are working on. So to conclude, we have designed a quantitative model of temozolomide, the pharmacokinetics and pharmacodynamics. And we have calibrated the model using several, numerous datasets. This model was used in silico to identify an optimal drug combination that was temozolomide plus an inhibitor of base excision repair plus an inhibitor of homologous recombination. Then using machine learning, we were able to identify the most important mechanisms that would need to be checked in order to decide to give this drug combination to patients. And perspectives, as I was mentioning, is to basically supplement the mathematical model with other pathways in a patient-specific way. And this is what we are doing using time-resolved proteomics under temozolomide exposure and drug screening. But this is another project which I will come back to tell you, to expose another webinar. Thank you very much.
Thank you very much, Annabelle. So for the sake of time, I would say we can take one very important question.
Basically, there was one question about how you set up your model. I think you feed your model with experimental data with published results. But what if in the literature, the published results are quite different on different experimental models? How you feed your model then?
Yeah, that's really a good question. So, of course, the model is a bit flexible in the sense that so what we have done for I will just go back to the slide. Okay, what we have done to try to constrain a bit the things that for about half of the parameters, they were basically set up on any the information was taken for any any cell line models, or because it was available. And then here it's true that some parameter those parameters are related to the biology of this cell line. And the way that we usually do that, in the sense, the way that we usually personalize the model is to look for the most important parameters or the most sensitive parameters, and then calibrate only those parameters to the new data. Meaning that basically we have the same model structure, part of the parameters are the same, and we just modify some parameters for each cell line, or in our case, at the moment we are using patient-derived cell lines. So basically the short answer is partial recalibration of the model.